When it comes to working memory, I think most of us would agree that individual memories vary in their quality, whether we call that memory strength or memory precision, doesn't really matter. And the current state-of-the-art procedure for measuring that quality is the continuous reproduction task. You're all familiar with this task, and the kind of measure that we obtain from it is the angular distance between the response and the target feature. Now, even though on each particular trial this is a continuous measure of performance, it's not a direct measure on the underlying memory trace, rather it's a proxy for this. And this is because current models of working memory assume that we do not encode color or orientation as a particular value. Rather, we encode the distribution of possible values via some kind of a population code. And it's the properties of that distribution that determine how precise our memory is. Thus, in order to estimate memory precision, one needs to measure hundreds of trials to estimate the underlying distribution, and then fit your favorite measurement model and extract its precision from that. This is the best that we can currently do, but there are a number of issues with this procedure. The first one is practical. As I said, you really need hundreds of trials in order to get reliable estimates of the underlying distribution. Of more interest to me is the second conceptual issue here, and that is that with this procedure, you cannot really tell anything about the quality of a particular memory. You can only tell about memory precision in the aggregate. And for the purposes of this talk, I will assume that this is something we as a field want to be able to do potentially. So how can we do better? Imagine the following procedure. After people provide a specific response on the color wheel, we also ask them to provide a range of values that are consistent with what they remember about the stimulus. And let's call this procedure uncertainty ratings. We can measure memory precision, hopefully, by measuring the width of this arc. Now, turns out I wasn't the only one to think of this procedure. It was introduced 3 years ago and has recently been used in a number of papers. But importantly, all of these papers have used it to study particular scientific questions about metacognition and have not investigated whether this is a good measure of memory precision and whether it's a better measure of memory precision, which is what I will do in this talk. I will address several questions today. The first one is, can people actually do this? What does the data look like? And does this new measure relate to existing measures of memory precision? If it does this actually solve the aggregation issue? Is this procedure better? Can we get away with making fewer observations while achieving equivalent measurement precision? Finally, if people can do that, and it's a good measure of memory precision, what are the psychometric properties of this new procedure? Is it actually measuring the same latent variable as established measures? I will show you results from 2 steps. In the first step, I will show you a reanalysis of existing data addressing the aggregation issue. And in the second step, I will show you results from a psychometric study, which was performed by an intern in our lab, Hui Chao Ji. She's currently looking for PhD positions, and she's fantastic. So if you see her application, please give her a consideration. The first data set that I've looked at comes from a recent paper by Hyonik and colleagues. In this paper, they asked participants to remember 4 colors and then to reproduce one of them on the color wheel. And in addition, they asked participants to draw an arc with the width of the arc reflecting how uncertain they are in their responses. Okay, so how well do these 2 measures relate to one another? From this task, we obtained 2 types of distribution. One is for the angle error, typical distribution centered around 0. And we obtained a distribution for the width of the arc. It shows a typical long tail distribution similar to reaction times. If we correlate on each trial the angular error and the arc width, what do we get? In several different studies, so in Hyonik et al., this was a correlation of about 0.30. In the other studies, this has varied from about 0.16 to 0.5 as well. So the correlation between them is weak to modest at the best. This is not particularly surprising because, as I said, the angular error for individual trials is just a sample from a distribution and doesn't necessarily reflect memory precision for that particular trial. So can we get this estimate a little better? In the first three analyses, what I did was to take each of the 12 subjects and split their data into each of the 4 sessions, so we get 48 distributions. And from those to extract 2 types of memory precision, one from the angular error using typical methods, and the other by calculating the mean arc width for each subject and session. So how do these 2 measures relate to one another? When we do that, we see that we now obtain a much higher correlation. So here on the x-axis, we have precision estimated from angle error and precision estimated from arc width for each of the 48 data points, and we have a correlation of about 0.8. Okay, so it looks like this new measure of precision is strongly related to existing measures of memory precision. But so what? Do we actually gain any new information from using it? What we have to remember that any experiment that we perform is just a sample from a population. And when we run multiple experiments, we might get different estimates of memory precision. And the variance in those estimates is our measurement noise. So we can try to estimate how much measurement noise this new procedure has relative to the existing procedure, and whether we can get away with making fewer observations achieving the same measurement noise. In order to do that, I run a bootstrapping simulation. From each participant, I drew a random sample of 300 observations. And for those 300 observations, I calculated 2 separate measures of precision from the point responses and from the arc width. Then I repeated that 1,000 times, and I calculated the confidence intervals of each of those 2 measures. The relative width of these 2 confidence intervals will tell us whether the new procedure is actually better than the old procedure. And here are the results. On the x-axis, we have 12 different participants, and on the y-axis, we have estimated memory precision. We have 2 types of points for each participant. In black, we show memory precision from the continuous reproduction task and the corresponding confidence intervals. As you can notice, for each participant, they're fairly wide. In contrast, precision estimated from the arc width, first, it's consistent with precision estimated from the point responses, but it also has much narrower confidence intervals over bootstrap samples. In fact, memory precision from this new task has about 7 times less measurement noise. This sounds almost too good to be true, but also a little abstract, so let's put it into context. In a typical continuous reproduction task, researchers usually gather about 2, maybe 300 trials. So let's ask the question this way. How many trials of arc widths can we gather that achieve equivalent measurement noise as 300 trials from the continuous reproduction task? To answer this question, I've done a bootstrapping simulation like the one before, but this time gradually increasing the number of trials from the arc width from 1 to 300. When we do that, on this graph, on the x-axis, I have the number of bootstrap samples, and on the y-axis, I have measurement noise. The red line is measurement noise from the continuous reproduction task from 300 trials. And we see that these 2 lines intersect at about 9 trials, which means that with only 9 trials of arc width measurement, we achieve equivalent measurement noise as 300 trials in the continuous reproduction task. Importantly, with such few trials, we still maintain very high correlation in the aggregate between precision estimated from the 2 procedures. This is great, but can we understand how well this new procedure works? This was the goal of the new experiment. First, we wanted to manipulate difficult gradually within each individual in order to obtain their mapping function from arc width to memory precision as estimated from the continuous reproduction task. And we wanted to test whether these mapping functions transfer from one type of experiment to another. And finally, we wanted to evaluate the psychometric properties of this new procedure. In order to establish that this new procedure measures the same latent variable, several factors need to be demonstrated. First, both procedures has to be affected similarly by typical working memory manipulations, such as set size, encoding time, retention interval. Second, within each participant, we want to be able to see that greater angle error is associated with less certain responses, but also the same has to be true across participants. Less precise participants have to give wider arc responses. Then we want to establish whether this mapping between the two procedures is stable. Does it transfer from one type of experiment to another? Finally, are the two procedures affected by similar biases? Today, I'll focus on the first four questions, but I'm happy to answer questions about the fifth one as well. Okay, so we've run two experiments, one set size experiment and one experiment in which we manipulated encoding time and delay. But importantly, people could perform either two set size experiments, one after another on different days. Alternatively, they could do a set size experiment followed by a time experiment, or they could do a time experiment followed by a set size experiment, or a time experiment followed by a time experiment. So that would allow us to test how well the mapping function transfers between different types of manipulations. In the set size experiments, people saw a display of 1 to 8 items, and then after 1 second, they had to reproduce it and give an arc width. In the time experiments, participants saw only one item, and we manipulated independently how long the item was presented for and how long people had to maintain it into memory. Okay, are these two procedures affected similarly by these manipulations? When it comes to set size, we can see here on the left point response memory precision and arc width here on the right. Both functions increase similarly as a function of set size. And in fact, if we plot them such that each point is a certain set size condition, and on here we have the angular error precision, and on the y-axis we have the arc width, we can see that these two measures are almost perfectly linearly related to one another. When it comes to the time experiment, we see exactly the same. So here on the x-axis, we have encoding time, and the colors represent different delay intervals. So both the point response's angular error and the mean arc width follow exactly the same patterns as well. And once again, if we plot these conditions for the two measures, we see a strongly linear relationship. Okay, so overall, the two measures are affected by the same manipulation. But if we look within each participant on a trial-by-trial basis, are these two measures associated? Indeed, this is exactly what we see. So here on the x-axis, we have arc size for individual trials, and we have the angular error standard deviation on the y-axis. What we could see is that we have almost a one-to-one relationship up until arc size reaches 90 degrees, which is half of the circle because we measure half of the arc. And afterwards, this relationship breaks down, possibly reflecting guessing responses. And if we run a regression analysis within this region, we can see that the mapping slope is almost perfectly one between the two procedures. Furthermore, we can see that if we look across participants rather than within participants, we see that the two measures increase concordingly as well. So people are using similar mapping functions. So finally, is this mapping stable between the two different experiments? First, look here on the left for people who performed both set size experiments on different days. We can see that the mapping from angle difference to arc size is perfectly reliable from one experiment to the other. The two lines fall exactly on top of each other. Similar finding is obtained for the time experiment, although there's a little bit more noise because the scale is narrower. If we look within each individual participant, we can also see that this mapping is fairly stable from one experiment to the other as well. The interesting thing is what happens when you change the type of experiment from one day to the other. Now we can see that even though the slope of the mapping function between the two experiments is similar, we have a huge difference in the intercept of this mapping function. Particularly if we look at set size 1 in the set size experiment, people are giving much less confident responses relative to set size 1 in the time experiment. I have an explanation for this, but I do not have time to get into it, but I'm happy to answer questions. In summary, we have learned two things about this new task. First, we have fairly strong evidence that it's measuring the same latent variable as the continuous reproduction task, but more importantly, it has orders of magnitude less measurement noise, which means that with only 10 trials, for example, we can achieve equivalent measurement procedure as 300 trials in the continuous reproduction task. This brings us closer to the ability to measure memory quality of individual items, and even though we cannot do that quite yet, I have some ideas and I'm happy to answer questions about them. Thank you for your attention.